everyone, I am Olumide Afolabi, and I work in the Biotech Department of Central Research and Diagnostic Laboratory, Tanke Ilorin. In this section of the channel, I'm going to be taking us through the protocols in the Biotech Department, and this cuts across DNA extraction, RNA extraction, gel electrophoresis, and basically everything that revolves around molecular biology laboratory analysis. Today we are going to be running some Agarose Gel Electrophoresis. Electrophoresis is a procedure in biological sciences for resolving biomolecules such as proteins, nucleic acids. And it basically works with two principles. So it resolves molecules based on their sizes and then based on their electric charges. So for nucleic acid electrophoresis, which is what we are going to be doing today, we are going to be resolving PCR product, which is DNA molecules based on their sizes. So DNA molecule is negatively charged, so we expect it to move from the negative side to the positive side on the electrophoresis machine. So, and before we go into the details, I'm going to give a brief outline of our protocol for today. So we are going to prepare an Agarose gel. So then after preparation of the Agarose gel, we're going to load our DNA samples, our PCR products on the Agarose gel. Then after that, we visualize the result. So I'm going to run us briefly through the materials and the materials in terms of reagents and the equipment required for this process. So we have a horizontal gel electrophoresis system, so which comprises of our gel tank, then, or some people call it gel box. So then our gel tray, and then the power supply unit. So, and here is the agarose powder we're going to be using. So this is from Excelris. We are going to be using agarose from Excelris, and then we are going to be using a buffer, TAE buffer, Tris acetate EDTA. So you don't have to use TAE. There are other buffers you can use, and one is a TB. You can use TB, which is Tris borate EDTA. I'm going to be preparing a 2% agarose gel for PCR product. So, and the process involved in this can be broken down into 3 stages. So the first is the preparation of my gel. So the second is the loading of the sample, preparation of the sample for loading, and then the loading of the samples on the gel. Then the third is visualization of the gel on the blue light trans illuminator, which I'm going to be showing us afterwards. So to prepare the gel, I'll set up my tray. So I'm going to be pouring the gel inside this tray, so that means I need to damp the ends of the tray. So I could use a rubber dam, or I could just use a tape dam. I'm going to be using a tape dam in this video. My tray is ready, so the next thing is for me to prepare the gel. Like I said, we are making 2% agarose gel. So, and the capacity of this tray is about 50 ml. So I'm going to be making 2% of agarose gel in 50 ml of buffer. I'll measure 50 ml of buffer into a flask, 1x TAE buffer, 2% of 50 ml is 1. So I'm going to be weighing 1 gram of Agarose into 50 ml of TAE. I'm going to be weighing 1 gram of Agarose powder into 50 ml. So I have my 1 gram. I pour that into the TAE buffer. So I swirl to dissolve. So the next step is I take this into a microwave oven, then boil the buffer until all the particles dissolves. So the gel is back from the microwave. So and usually the microwave step takes like 1 to 3 minutes. All you just want to, depending on the temperature you set, what you want to check is whether the buffer and the powder has totally dissolved in the buffer. Now it's very hot, so I cannot proceed to the next stage, which is adding of the DNA stain to the gel. So to hasten the process, I can cool under tap. So but if I don't want to cool under tap, I can just place this on the bench and allow to cool down. So I'm going to cool this under the tap. Right now, so you don't want to do this too much, because if you do, the gel just gel up, it solidifies before you are able to pour into the tray. So the next step is to add the DNA stain. The stain I'm going to be using is ethidium bromide. So, and you have to be careful when you're using ethidium bromide because it's a suspected carcinogen. So the concentration of ethidium bromide I'm adding to a final concentration of 0.5 microgram per ml. So our ethidium bromide is already diluted to 10 milligram per ml. So I'm basically going to be adding 2.5 microliters of ethidium bromide to the gel. So I'm adding 2.5 microliter of ethidium bromide to the gel. So remember, the concentration of my ethidium bromide is 10 milligram per ml. So I'm just going to add 2.5 microliter of that to my 50 ml gel. Then I swirl gently to mix. After mixing, I pour the gel into the tray.
Because the organism means the nano didn't work on this. Unlike this, the organism is high here, and the organism is.
Then I check the gel to be sure there are no bubbles on it. If there are bubbles, I can remove that with the tip. So it looks like I'm fine, no bubbles on the gel. Then I set my comb. The comb creates
So this one
it creates wells in the gel where samples will be loaded. So I set the comb in the gel.
Yeah, but here I did not pick it.
So, and that's the gel preparation. I wait for the gel to solidify, and when the gel solidifies, I load my sample, and we are ready to run.
But we still
So the second set of activities or protocol for the gel electrophoresis is to prepare my sample and get them ready to load on the gel. So, and the major thing I want to do is to mix my sample with a DNA loading dye. Now, the loading dye, the purpose of the loading dye is to give the DNA bulk so that it sinks to the bottom of the well when I'm loading, and to also serve as a tracking dye. Like, I know how far my sample has moved in the gel by merely looking at the migration of the dye. This is a 5x dye, so being 5x, I add 1 microliter of the dye to 4 microliter of my DNA sample. So the DNA sample I'm working with, they are PCR products. So I've just run some PCR and I want to load them on the Agarose gel to see whether my amplification was successful. So we might not really get like some bands from this because this is a product from a PCR optimization that I'm working on. So, but I'm going to load some ladders alongside that. I'm going to load a 1 kb ladder and an 100 base pair ladder alongside my sample so that we can use those as a contrast. So now I'm going to put 1 microliter of loading dye in each tube before adding my sample. One microliter. Yeah, this is how the 1 microliter looks in my tube. And by the way, the tube I'm using now is a PCR tube, which is 0.2 mil tube. So after that, the next thing is I'll add the samples to the loading dye. 4 microliter of my DNA sample, that's my PCR product. So before I do that, I have to vortex and spin down this product before adding it to the dye, and I'm going to do that just now. I'm going to vortex the sample and spin down. After vortexing the sample and spinning down, I add 4 microliter of the sample, of each sample, to the loading dye in the tube. So then I spin this down again on the centrifuge, and then my sample is ready to load on the gel. Spinning. Next step is to load samples on the gel. By now our gel is solidified, so we peel off the tape used to damp. Then we place the tray containing the gel into the tank and fill the tank up with 1x TAE buffer, the same buffer that we used to prepare the gel. So we ensure that the gel is submerged by the buffer. After submerging the gel with the buffer, we remove the comb gently. So the comb has created wells, which may not be very visible because of the white background. It has created wells in the gel, and it's inside these wells created by the comb that will load the samples we have prepared. So I set my micropipette to 5 microliter to pick up the total content of the tube, which is 1 microliter plus 4 microliter of sample, 1 microliter dye plus 4 microliter sample. So I pick the 5 microliter, then I gently load in the well. So like I said, I'm going to be loading some ladders along with my sample. Okay, now I'm done loading. The next step is to set up the electrophoresis machine and connect it to the power supply unit and run. So I'm going to be running at 70 volts for 45 minutes. So 70 volts for 45 minutes. The time for running is 45 minutes. now the machine is running. So this runs for 45 minutes, then I check under the trans illuminator. Our run has concluded, so we can see that the loading die has migrated from where it started, from where we loaded initially. So it has migrated, like almost getting to the middle of the gel. So if I want further separation of my DNA, I would really allow this to run beyond 45 minutes. But for the purpose of my work, this is just fine for what I want to do. So then I remove the gel from the tank along with the tray and transfer to the gel documentation system. So the gel illuminator or gel scanner that we use is Lycor D-Digit. So I'll gently transfer the gel to the gel scanner. Make sure the gel is straight on the gel scanner, then remove bubbles from under the gel. So then I close that. So on the system, I locate the icon for the gel scanner, which is D-Digit here, then double click. So when this open, I adjust the exposure time. So we have optimized this already. So we had to take series of pictures using different exposure time to see which works better for the dye we are currently using, which is ethidium bromide. So if we switch to another dye, another DNA stain, say gel green, we have to re-optimize this to know which exposure time works better for that. After setting the exposure time, I click on the image capture to take the image. Okay, the capture is done. I have my images. I have my capture ready, and I can crop to just the region of interest. So capture to just the region of interest. So then I can view that on a convenient image viewer, say Windows Photo Viewer, where I can rotate and actually see my gel. So this is my result. Is not really looking good. Just like I said, I'm trying to optimize this PCR to see what would be the best condition for the PCR. So basically it looks like my DNA is stuck in the well. So a lot of things can be responsible for that, or maybe there is just some other contaminant that are stuck in the well, which are the bright spot in the well here. So it could just be that the concentration of my PCR product is just too high. One thing I might want to work with is probably reduce the number of cycles I'm working with. So, but just like I said, I added DNA ladders, so which is a test of sanity to give some sense of sanity that my gel was fine. So the first lane here is 100 base pair, and this lane, second to the last lane, is still 100 base pair, and the last lane 1 kb ladder. So ladders are standards, like DNA standards, with different sizes of DNA, so that when you load them on gel, they resolve to their according to their sizes, and you have something that actually looks like a ladder. The first band here is 100 base pair, second 200 base pair, 300, 400, 500, 600, 700, 800, 900, 1000, 1005, 2000, and then 3000 base pair. That's for the 100 base pair ladder, so which is also applies to this. So but for the other ladder, which is 1 kb ladder, so it starts from 300, and we can see that this almost aligns with the 300 of 100 base pair. 300, then 500, so it aligns with the 500 of 1 kb, and the other orientation band here, this stick band, is 1000, and the graduation goes up till I think 12,000 base pair. Yeah, that is our run for today. So we have successfully run an agarose gel for a PCR product we are trying to optimize our PCR conditions. So, and we have run the product on gel, and through the process we've seen how to make 2% agarose gel, how to prepare samples for loading, and how to eventually run the gel and then visualize on the gel documentation system. Thank you.
Don't forget to like, share, and subscribe so you don't miss the next video.